Mercury occurs in the environment as metallic mercury, and mercuric sulfide. The toxicity of mercury came to the limelight after the incidence of Minamata disease in 1953 to 1960 in Japan. So, among that, 1,111 cases of mercury poisoning were reported among the people who had eaten the mercury contaminated fish from the Minamata Bay. So, among them, about 45 people they have died. Genetic defects were observed in 20 babies whose mothers had consumed seafood from the bay. The sea fish in the bay were found to have 30 to 100 ppm of mercury in the form of methyl mercury. This is mercury source was the effluent from vinyl chloride plant of Minamata Chemical Company, which is discharged into the bay. Another tragic report of mercury poisoning from Iraq in 1972, where 450 villagers have died after they were eating the wheat which has been dusted with mercury containing pesticide. These two tragic events boosted the awareness of mercury as a pollutant, resulting in its being studied more extensively than any other trace elements. The sources of mercury are ore of mercury is cinnabar HGS, fossil fuels, coal, and lignite contain about 100 ppm of parts per billion of mercury. The natural abundance in the soil is zero point 0.1 parts per million. Toxic effects of mercury. Elemental mercury, that is Hg non-toxic in nature, and mercurous ion Hg2 2 plus R2Hg low toxicity. Hg2 plus is toxic but not easily transported across the biological membrane. RH plus or CH3Hd plus organomercurals, it is highly toxic in nature that causes the irreversible brain and nerve damage, easily transported across the biological membrane and stored in the fat tissue. Symptoms of mercury toxicity: central nervous disorders, headache, irritability, fatigue, inability to make decision, sleeplessness, and diarrhea. Biological methylation are in the food. Mercury or its compound. Biological methylation or amplification in the food chain. Mercury or its compounds and anaerobic bacteria are responsible for the synthesis of methane in water. The conversion is facilitated by cobalt 3 containing vitamin B12 coenzyme. The methyl group present in the cobalt vitamin B12 is transferred enzymatically to mercury as a methyl mercury, as shown below. So it is vitamin B12 reacts with the mercury metal in the presence of methyl cobalamin and ATP. It gives rise to methyl mercury. Methyl. Thus, methyl mercury produced enters into the food chain along which its concentration increases to produce bioamplification. In the fish, the concentration is increased by a factor 10 power 3 or more. Hg2 plus biomethylation, anaerobic bacteria gives rise to methyl mercury. Plankton, that's go next, that is transported to small fish or the birds, then it will go into large fish. This is how the biological methylation or amplification in the food chain. Treatment of mercury toxicity are the antidotes for the treatment of mercury. Chelating agents such as British anti-levisite can be used, or calcium sodium salt of EDTA, or N-acetyl penicillamine can be used, CH2S, CHSH, CH2SH. So this is the structure of British anti-levisite or 2,3 dimer captopropane 1 all.